Good evening to everyone present here. I am Gurupriya R, Prime Minister Research Fellow in the Department of Civil Engineering, IIT Madras. So I'll be handling the live sessions for Applied Environmental Microbiology course in the NPTEL module. So today is the fifth live practice session for this course. We'll get started with the session. I guess I'm audible. Uh, Can you please respond in the chat if I'm audible and if the PPT is visible? Okay, thank you. So in the week 5 course content, everything related to microbial genomics was dealt. We will solve some questions related to microbial genomics, and in between we will also discuss about that concept. Before that, is there any doubts from the previous lectures or previous live course live sessions? If there is some doubt, you can put in the chat box. I'll wait for 2 minutes. You can also unmute yourself and ask your doubts if you have any. Okay, I guess there are no doubts from previous sessions. In between also, if you have some doubts, you can ask me. So now I'll present the first question for the day. You can try answering. You can give the answers in the chat box. Okay, I see no responses as of now. SBS approaching. It is not SBS approaching. Um, yeah, based on dideoxy only, there is one name for the method. That's okay. We have many more questions to solve. Now, let me tell the answer for this. It is known as chain termination method because the binding of this dideoxynucleotide will terminate the chain. Like we cannot add furthermore nucleotides to that uh, chain. So that is known as chain termination method. I'll now show the display the second question. You can try answering it. I think this is a very easy question. I think this is a very easy question, and probably for the last question, the answer was told here. So let me just display the answer here. So we use both deoxyribonucleotides and dideoxyribonucleotides. Can someone say what's the difference between deoxyribonucleotides and dideoxyribonucleotides? Basically, DNTPs and DDNTPs. You can just mention which position, what's the change. So you can respond to the chat box. It may be right or wrong, that doesn't matter. You can just interact. Anyone who knows the difference between a DNTP and a DDNTP? Right. DNTPs are nucleotides and they are building blocks of DNA. You're perfectly right. Okay, let me tell the difference between DNTPs and DDNTPs. So, as Salakeshwari has told, DNTPs are nucleotides and they are building blocks of the DNA. Structurally, they have a ribose sugar. So, there is a pentose ring. In that pentose ring, in the second position, instead of H and OH, H and H are present. Instead of H, there is instead of OH there is H. So that is why it is called deoxyribose. There is no that the O is not there, so that is why it is called deoxy. Okay, whereas in the case of DDNTPs, dideoxy means in some other position also the OH is replaced by H. So that is the third position. So basically in DNTPs, we have the deoxy group in the second position. In the DDNTPs, we have deoxy in both second and third position. So that is the structural difference between DNTPs and DDNTPs. Okay, so that is why when the chain elongation is happening in the third position, we are not able to um, do any attachment. So generally we read the DNA sequences from 5 prime to 3 prime, right? So in the 3 prime end, because hydroxyl is present, there is we can form the bond for the next nucleotide to come and join there. Whereas in the case of DDNTPs, in the third position the hydroxyl is not present, so the next nucleotide cannot come and bind, and in that with that position itself the chain elongation is stopping. So that is why this is called a chain termination reaction. Okay, right. So I guess it was clear. If you want me to repeat, you can put in the chat box so that I'll repeat. Was it clear or should I repeat? Can I get some response? Okay, right. So I'll display the third question. Again, this is also kind of a warm-up question. If you know how Sanger sequencing is done, I'll give you some time to think and answer. So in the case of Sanger sequencing, we are doing a DNA elongation. So you can just think which enzyme we have learnt about in the DNA elongation part. Maybe in the first few weeks, we studied about transcription, translation, replication, and all. So in the central dogma, DNA polymerase enzyme. Okay. Any more responses? Okay, DNA polymerase is perfectly fine. That's the answer. So do you know what are the steps involved in Sanger sequencing? Because that's the very first DNA sequencing method, and it's very important to know how uh, that is being carried out. So, do any of you know what are the steps? Roughly, some three steps or something. Okay. Any idea about Sanger sequencing? Okay, let me explain about Sanger sequencing then. So yeah, as this picture here suggests, there are three steps in Sanger sequencing basically. Okay, so and the method involved, as we saw, is the chain termination method, and here we carry out the PCR as the first step. So this PCR is known as chain termination PCR. In this, we have um, addition of a low ratio of modified nucleotides called DDNTPs or the chain terminating nucleotides along with the normal DNTPs in the PCR reaction mixture. So in the reaction mixture, in the PCR reaction mixture, we should be adding the nucleotides. Generally, in other PCRs, we will be adding only DNTPs because we just need multiple copies of the DNA. Uh, so we'll add normal DNTPs. But in the case of sequencing, what we do is we also add a smaller ratio of dideoxyribonucleotides. As I told you, there is a structural difference between DNTPs and DDNTPs, which allows DNTPs to continue um, nucleotide addition, whereas in DDNTPs it is terminated at that point itself. Okay, so this DDNTPs lack the 3' hydroxyl, third position hydroxyl group required for the phosphodiester bond formation. So once this DDNTP is incorporated, the DNA polymerase cannot extend uh, the chain further. So after PCR, we get millions to billions of copies of oligonucleotides or, or the DNA sequences that are terminated at random lengths. As you can see in this picture, we have this original strand. We have that has to be sequenced. So we have added a mixture of DNTPs and TDNTPs, which are fluorescently labeled. Okay, so once this at different positions we can see that different DDNTPs. For example, when a DDNTP of or a DDGTP is bound here in this position, corresponding to this position, it will stop here. Similarly, here it is going to be TTP. So like that we will get different millions and billions of copies that are terminated at different lengths. So once this is done, what we are going to do is we are going to do a gel electrophoresis. So have you heard of gel electrophoresis? Any of you has heard of gel electrophoresis? Can someone tell what's the uh, principle of gel electrophoresis? Just in a sentence, what drives gel electrophoresis basically? In our case, what drives DNA gel electrophoresis? Have any of you heard about gel electrophoresis? You can just tell a yes or no. If yes, you can tell me what is the driving force, or else you can, yeah. Can you just tell me what's the driving force in a gel electrophoresis, or what principle it's based on? Okay, yeah, we use it to separate DNA fragments based on. You're perfectly right. We are going to separate the DNA fragments, but based on what criteria are we going to separate them? That is basically the principle. Okay. Um, Let me explain that also. So, as she has mentioned, gel electrophoresis is basically a technique used to separate DNA fragments of proteins, anything. Okay, so in our case it is DNA gel electrophoresis. So we are going to separate DNA fragments. Electrophoresis, as the name mentioned, is we are going to apply some electric or potential difference, electricity basically, and then allow the DNA fragments to move. So when we are allowing them to move, what happens is this gel is kind of a matrix. We have agarose gel electrophoresis, for example. So in that one, what happens is we just give a gel matrix which is kind of porous. So through that spaces or pores only our DNA is going to move through. So once this is moving, mm, based on its size it will be stuck at some point, uh, right? Because these pores are not going to be of uniform size. Whichever is the smallest fragment will just move till the last very faster, whereas the heavier fragments, so the larger fragments are not going to force as much as the shorter fragments are going to do. The driving factor here is DNA is negatively charged. So when we are going to apply a potential, uh, the DNA fragments are going to move towards the positive electrode. Uh, so they are going to move towards the positive electrode, and based on their size, they will be stopped at different positions, giving us different bands. Okay, so with those bands we can just identify that will give us some information. So in this case what happens is we are going to do the chain termination PCR, so that we will get different lengths of DNA fragments like this mentioned here, and then we are going to perform the gel electrophoresis. Okay, so size separation by capillary gel electrophoresis. The larger fragments are going to stay up and the smaller fragments are going to come down. Okay, so and then the DNA polymerase only synthesizes DNA in the 5 prime to 3 prime direction, starting at a provided primer. So like in the case of replication, we will require a primer here, and then from that primer 5 prime to 3 prime directional addition is going to happen, and each terminal DDNTP will correspond to a nucleotide in the original sequence. And after this electrophoresis happen, we are going to take the gel and read the gel. So as we have mentioned here, we have a fluorescently labeled DDNTPs. So we can read it using that fluorescence principle. It is going to emit different lights to us. So based on that, we will know if A is there in that position, or T is there, or G is there, or C is there, based on different fluorescent labels. Okay. So based on that, when we are going to pass that light on them, we are going to get different signals. As mentioned, if it is going to be this blue color, you call it C. If it's going to be red color, you call it T, something like that. So that is how you read the gel basically. By reading the gel bands from smallest to largest, we can determine the 5 prime to 3 prime sequence of the original DNA strand. So this way, when you are going to read it one by one, this is going to be your 5 prime to 3 prime of that DNA strand, which for which you are performing the sequencing. So in Sanger sequencing, I'll just sum up. We have three steps. The first step is the PCR. So in PCR, what we do is we take the original strand, we add all the required PCR mixture. In this mixture, the important point to note is we are going to add DNTPs along with DDNTPs. So when DDNTPs are going to bind, what happens is the chain is going to stop. The chain elongation is going to stop. So we get millions and billions of copies of different DNA fragments of different lengths that has been terminated at different points. So that's basically an advantage for us to run the gel electrophoresis. So the larger fragments will be near the sample well itself, whereas the smaller fragments are going to travel further down. So after that, what we are going to do is we have already fluorescently labeled the DDNTPs. So we can read the gel for those fluorescent signals, and based on that we can determine the 5 prime to 3 prime sequence of the original DNA strand. This is how Sanger sequencing is performed. Is it clear? Okay. Others, it's clear for others also? Others, it's clear or should I repeat? Okay, let me ask one more question. So I see some three, four people apart from me here in the meeting. Are you all from some biotechnology, microbiology kind of a background? Or because for the past few weeks, the audience I had were from biotech background. So is it the same this week also? If you are from some other backgrounds, it's not an issue. I can explain things from scratch. Okay, you're from civil engineering background. So, did you understand whatever I told till now, or should I explain you from scratch? Okay, you're from nutrition. So, you people can let me know if I have to explain you what a DNTP is, DDNTP is, how a PCR works basically. I can do that. Is this fine, or should I explain it that way? You can just put it in the chat box so that. Okay. Okay, fine. Maybe with the next, we'll proceed with the next question. And if you have some doubts, if you want me to repeat, you can let me know. NGS consists of dash steps in sequence. If someone has heard about NGS, what's NGS? So have you heard about something called the next generation sequencing? Yeah, next generation sequencing. So NGS stands for next generation sequencing. The Sanger sequence, we call it as the first generation. That's very basic and that's very time consuming. So, but that was the first discovered method for DNA sequencing also. We have multiple methods and we have few techniques called the next generation sequencing, and we have a common methodological scheme for it. Are you aware of it? This was also briefly discussed in the lecture videos of week 5. Even when not in sequence, you can just tell if you remember any of the steps in the NGS sequencing, in the NGS basically, next generation sequencing. Okay, that's completely fine. Oh, you're technically right. Data analyzing is, but technically this would also be the last step. So first you have to generate your data. There are few steps for generating your data, and finally, what you do is analyze the data. Ah, library preparation. Yes, there is library preparation. Great. So library construction, amplification, sequencing, and finally what you do with the sequence is data analysis. So this is how NGS works. So in whatever methodology you're going to work with, you will first construct a library. You are going to perform the gene amplification using uh, different PCR. So having said that, let me first tell what a PCR is. PCR is basically polymerase chain reaction. Okay, polymerase chain reaction is a technique widely used in the biological field for DNA amplification. So as the name suggests, amplification is making multiple copies. So I have a DNA fragment with me. In order to study, uh, in order to study that, I will be needing multiple copies, some thousands of hundreds of thousands or lakhs of copies even, uh, for analysis, right? So what we do is we have a double stranded DNA, and we what we do are going to do now is we are going to amplify it to multiple copies. For that, the technique that we are going to use is known as polymerase chain reaction. As the name suggests, we have studied in the central dogma, right? Polymerase, DNA polymerase is the enzyme which we use for making the DNA strand or the DNA chain. Okay, so we are going to use the same polymerase enzyme and going to create the DNA chain. So that is why it is called a polymerase chain reaction. Okay, so in that reaction mixture, what you will be having is different components. So as we saw in the Sanger sequencing, the nucleotides are the very major component for PCR to happen. First, what happens is you put a double stranded DNA in the mixture, and that has to be first separated. The two strands will be separated. We put in a double stranded DNA, give some heat. So we know heat is going to denature our DNA molecule. So our two strands will be separated. And after that, another component of the PCR mixture is the primer. In the central dogma again, we have learned that whenever we are going to perform, for example, DNA replication or something, there will be some primer to it. That will be the first few sequence or first few nucleotides that will be attached to the complementary strand, and from that side only DNA polymerase can function. Just a minute, let me take up a pen. So what happens is we have our double stranded DNA like this. So when we are going to apply heat, what happens? We'll have a higher temperature basically. Apply heat in the sense a higher temperature of the mixture. What happens is these two strands are going to separate like this. Okay, these two strands will be separated. After that, what happens is the PCR mixture will contain a primer. So this primer will be complementary, for example, to this region of this strand. There are two strands, and if this is complementary, it will attach. So what happens is from here only our DNA polymerase will act. So here our temperature will be further reduced. Here we heated it up, right? So here we are going to reduce the temperature to some extent. So what happens is this, here we will not reduce the temperature, we will use something called a Taq polymerase. So a Taq polymerase is a Thermus aquaticus polymerase. Thermus aquaticus is an organism which is heat resistant. So we have studied about halophiles, psychrophiles, thermophiles, different organisms that can survive in different conditions, temperature, pressure, salt, pH, etc. Right? So in that, this Thermus aquaticus is a highly heat resistant organism. So the polymerase present in this Thermus aquaticus will be working in higher temperature, whereas if you are going to take it from a normal microorganism, it will not work in higher temperature, it will get denatured at higher temperature. So here we have applied heat, this has denatured. Okay, so we are going to maintain that kind of a higher temperature only, so that the strands do not reunite again. So in this from this primer, this DNA polymerase is going to again extend this strand. Okay, again extend this strand. So this will obviously be complementary to this. If A is present here, what will DNA polymerase do? It will bring T here. Okay, so after this, what happens is next step will be this DNA polymerase has done its duty. It will leave. Uh, Everything has done. So what happens is, next when we are going to reduce the temperature, these two strands will again be supercoiled, or the double helical structure will be again brought back. So what we get at the end is, we will get one more copy of the DNA here basically. So from this one strand we are going to get another copy of the DNA. So at the end of this PCR reaction, what you're going to get is multiple copies of DNA. So this is how PCR works, or this amplification step is working. Okay, I guess PCR was clear. So was PCR clear, or is there any doubt in it? Okay, so right. We will see those NGS steps in details in the future slides. Yeah, but this question, yeah, you can answer. Ion torrent semiconductor sequencing employs basically the principle. You can just tell one word or two, three words, whatever. You can just answer this question even in a word. If you have gone through the lecture videos carefully, this was discussed in the lecture videos also, the ion torrent sequencing. There was a key difference that was told between ion torrent sequencing and the other sequencing methods that we have seen so far. The detailed procedure of how ion torrent sequencing is done or it's being carried out is not much necessary. You just have to know the principle. What's the key difference between this method and the other methods? Okay, I guess this term is pretty new. So let me just tell the answer. Ion torrent sequencing is the one that basically works with the help of pH differences. So what happens here is, in our Sanger sequencing, what we did, we used fluorescently labeled DDNTPs. So at the end, what we did was, based on fluorescence principle, we did the gel reading or the detection. So we'll get different lights of different wavelengths when there are different fluorescent labels. For A it is going to be one color, T it is going to be one color, G and C they are going to be in different colors. Okay, so that is how we did the detection in our Sanger sequencing. Uh, Mostly optical methods were only used for this detection also. So in the case of iron torrent sequencing, what happens is it senses the difference in pH because hydrogen bonds will be formed. So when hydrogen bonds are formed, the free hydroxyl, the free H plus ions are not going to be in the reaction mixture, right? So our sensor is going to sense what is the difference in the pH. Basically, negative logarithm of hydrogen ion concentration or hydrogen ion activity basically. Okay, so that is going to be sensed, and based on that sensing it's going to give us signal, and with that signal we are going to have the read for our sequencing. Okay, so that's the main purpose of iron torrent semiconductor sequencing. It uses the pH differences to give us the reads. This is another question related to next generation sequencing. So which of the following does NGS employ? You can just guess some answers. I'll anyway explain what's the answer and why it is so. Think about what's Sanger sequencing and what will be the difference in the next generation. So roughly we have an idea that we have library preparation, amplification, steps. Okay, so based on these two steps itself you can just guess the answer, what will be the answer. Great. Great, and that's absolutely right. So we are just using the sequence by synthesis principle. So what we do here is we are going to synthesize DNA strands basically. In chain termination, it's not chain termination because chain termination we saw it in Sanger sequencing. Chemical degradation is not happening here. Uh, NGS employs amplification basically. So it cannot be none of the above. It's perfectly right. NGS employs sequence by synthesis principle. Okay. So in NGS we have heard about bridge PCR and emulsion PCR in the through the lectures. So this question is based on bridge PCR. Which bridge PCR promotes which of these? You can just imagine bridge PCR is basically you're going to have two things to which it's attached like a bridge. Okay, I guess this question is a bit difficult or beyond imagination or something. So it uses clusters of identical strands because, as the name suggests, we are going to have a bridge here. So obviously, one strand only is going to be attached. So we will have clusters of identical strands here. I think this question is pretty easy. This is the first commercial technique not to use fluorescence in camera scanning. To be more clear with the question, we are discussing about this sequencing methods, right? So I'm asking about that. Maybe just a few minutes back, I told the answer. Fluorescence in camera scanning, we basically do for detection. Okay, that's their detection techniques. Like in the case of Sanger sequencing, what we did, we had fluorescently labeled probes, and we will do a fluorescence scan to read the gel. Which technique does not use this fluorescence in camera scanning, as far as we know? You can just make a rough guess based on whatever we have discussed so far in all these previous seven questions. What could probably be the technique that doesn't use fluorescence in camera scanning for detection? We have seen about sequencing so far. So we just now saw one kind of sequencing that did not use fluorescence in camera scanning. That's the answer. Okay, I guess this question is also difficult. You want me to display the answer for this, or should I give some more time? Okay. Ion torrent sequencing again. We discussed about it few minutes back, right? So that is one of the techniques that does not use fluorescence. Because what did it use? It did not use fluorescence sensing or camera sensing or any optical methods. It was based on pH, right? You're not going to get any optical signals like based on some wavelength, some light or something like that. It's just based on hydrogen ion concentration, the pH. Okay, that was kind of a trivia question. The key enzyme used in pyrosequencing. Have you heard about pyrosequencing? This was kind of discussed in the lectures, and yeah, basically in any sequencing, DNA polymerase is going to be used, but there is one enzyme that's very specific to pyrosequencing, um, based on which this method was developed. This has nothing to do with your DNA fragment and its elongation and all. Okay, this is also used, but this is not the key enzyme. You're very near to the answer, but not exactly. Yeah, firefly luciferases. So you are pretty right with all the enzymes because all of them are used in pyrosequencing. Do you have an idea of for what ATP sulfurylase is used generally? Okay, ATP sulfurylase is an enzyme which converts APS to ATP. Okay, so when this ATP is formed, uh, the luciferase enzyme acts, which converts luciferin into oxyluciferin, giving also along with giving light. Okay, and then apyrase is another enzyme. It continuously degrades the uh, nucleotides and ATP. So basically, from ATP, the apyrase is going to give you ADP, AMP, and phosphates. But when you're going to be asked about the key enzymes in pyrosequencing, it's going to be luciferase. I'm sorry, the enzyme should be luciferase. When I upload the PPT, I'll correct this now. So the answer for this is luciferase. Okay, now we'll have some discussion. So as discussed in the NGS, what we have is library preparation. DNA is fragmented and ligated to specific adapters in preparation for amplification. So these are some adapters. So our DNA will be attached to these adapters. After that, what we do is we do the clonal amplification. So clonal amplification is basically uh, by either emulsion PCR on a bead or a bridge PCR on a solid surface. So after this is done, so this is how it looks. So this one is the emulsion PCR. You have some emulsion missiles or droplets kind of a thing to which your DNA is attached, and then there the PCR reaction is going to happen. The PCR that we discussed. So whatever I told about PCR is the general steps in PCR. You have multiple kinds of PCRs, each with a slight modification. Like in the case of bridge PCR, you're going to have two adapters here to which it's going to attach like a bridge. So it's called a bridge PCR. Here, what you have is an emulsion uh, bead to which your DNA is going to attach and get amplified. So this is called emulsion PCR. But basically, the steps of denaturation, annealing, denaturation, amplification, annealing, and renaturation is going to be same. Okay. And then what you do is after your PCR, now you have enough copies of DNA with which you can perform this sequencing. The output is recorded, and the fourth step will be data analysis. So during library preparation, the DNA is usually fragmented using some enzymes or physical methods and ligated to platform specific adapters. So based on your requirement platform, you have specific adapters to which it will be attached. And after that, the template DNA is amplified via a droplet or a emulsion PCR or a solid state or a bridge PCR in order to generate more copies for the sequencing. Then what you do is you do the sequencing based on optical signals, for example fluorescence, chemical signals, for example pH. So fluorescence, you have to remember about Sanger sequencing, that is using optical or fluorescence, and the chemical signals we have ion torrent sequencing which uses pH. um, so what happens here is, based on the addition of nucleotide to this DNA strand, there is going to be difference in this fluorescence optical signal or the chemical signal. That signal is going to be recorded, and that data you will be analyzing, and you will be telling, okay, this is going to correspond to this nucleotide. In this position, this nucleotide was present, and finally you get the final sequence of your DNA. What happens in emulsion PCR? So emulsion PCR is basically used in pyrosequencing method, proton sequencing, and sequencing by ligation. What happens here is, first the adapter ligated template, as we saw here. So this is the adapter ligated template, that the primer, the polymerase, and the beads are contained here in the missile. So this is one missile. So what you have here is an adapter ligated template. You have a primer, you have polymerase in this bead. Okay, the template binds complementary primers on the bead, and the PCR generates the complementary strand. So by this method, what you're going to do is you're going to multiply or make multiple copies of your DNA, and then the beads with clonal polonies are then used in the sequencing reaction. So you have the primer specific attachment of DNA, its multiplication, and finally you're going to take it for your sequencing. In bridge PCR, what's the case is, the adapter ligated template binds complementary primer on the surface. So this way, as it's shown here. After that, what happens is, the free end of the template binds the complementary primer, forming a bridge. Bridge PCR generates the complementary strand, and finally you can also denature the strands and form new bridges with free primers. This is repeated to form clone clusters. Okay, I see a hand raised. Do you have some question? I think it's Rakesh. Do you have some question? Yes, Alageshwari, you have some question? Yeah, you have some questions. You can either unmute and ask. You can also put in the chat box. I see two hands raised. Okay, I'll continue and I'll wait for your questions in the chat box. So I'll clarify as and when I see the questions. Okay, this is basically what pyrosequencing is. So instead of labeled DNTPs and physical separation of fragments, a two-step chemistry is used, and the detection is based on chemiluminescence. This is basically what this luciferase does. It emits light, and this phenomenon is chemiluminescence, uh, based on the presence of pyrophosphate that is being released. So what happens here is, we have a DNA template. Uh, there is going to be this nucleotide incorporation. So at the end, you will get this PPI, which will combine with APS to give you ATP. Here only, as she told, ATP sulfurylase is used, and this will again convert the luciferin can be converted to oxyluciferin by the luciferase enzyme, and light is emitted, and you get the pyrogram. This is how pyrosequencing works. So this is the next question. So in the lectures, we have seen about multiple studies, um, genomics basically. We also saw something called epigenome, right? So it's going to be related to that, epigenetics basically. What is epigenetics? Has anyone heard about it? No, it is not study of the environment. Genetics is basically you're going to study about genes, DNA, right? So epigenetics is going to be something related to that. Right. Environment change effect. You're perfectly right. So what we're going to study in epigenetics is basically the behavioral and environmental causes that change or affect the way the genes work. Excuse me. So what you're going to study here is you basically have your DNA. Okay, there are many things that can affect our DNA. That in epigenetics, what we basically study is how our environmental factor or our behavior is going to affect the DNA. Though this course is microbiology course, I'll just quote a simple non-microbiological example just to make you understand what epigenetics is. So basically, we are human beings and we live in different parts of the world. We have different food habits, we have different culture, and we have um, various diversities. So, for example, let's take a person A who constantly is in an environment which is very highly polluted with some kind of a chemical. Okay, the exposure, okay, the continuous exposure of the person to that particular chemical will obviously make some changes, can make some changes to the way his genes are working. Okay, whereas in other case what happens is let's say there is another person who is very healthy, both of them are healthy only, like the person A, another person B is also very healthy. He is also in the same place and being exposed to the same chemical, but there is no 100% guarantee that he will also, his genes are going to work like the person A. It differs from person to person. Similarly, in the case of microbes, what happens is epigenetic studies is very important because when you're going to expose the microbes to different kind of environments. For example, it can be a temperature factor, or it can be some chemical factors, it can be a food factor. Whatever it is, when you're going to continuously expose, there is going to be some changes in your genes. For example, if you have heard about DNA methylation, so DNA methylation is basically addition of methyl group to your gene that will turn off the gene. Turning off genes means we have studied the central dogma, where from DNA we get the RNA, and from that we get the protein from the transcription and translation process. So when we have a very active gene that encodes for a protein, um, this DNA to RNA, mRNA conversion, from mRNA to protein conversion happens naturally. Okay, that is how our enzymes or other proteins in our body are generated or produced. Okay, whereas in the case of epigenetics, what happens is sometimes that a particular gene may be methylated. So when it is methylated, it will not be expressed, so the gene will be off. That is what we call as epigenetics, study of epigenetics. So you have two rats which are of different colors due to some protein. One is in black color and one is in some blonde color or something. So because of some kind of a exposure to some kind of environment or food, one of it has changed its colors. For example, a normal gray color or black color looking rat changes color to a blonde color. You do a genetic analysis and you find the rats that are exposed to a certain kind of environment have some of the genes methylated, and because of that, those alone look in a different color. So all these studies is the study of epigenetics. It is very very important and a very crucial study, because we know we are being exposed to new things every day, and the same things every day. Okay, we are also exposed to new things, we are also having certain habitual exposures. So how that affects our genome, that study is very very important to have this interrelationship studies, or and other gene based studies. So the next question is also something similar. The analysis that is done to identify various microbes in an environmental sample. So for example, I'm going to give you some sample. For example, I took one sample from a lake, a water sample from a lake, and I'm just going to give you, I'm going to ask what are the microbes present in it, or give me a microbiological report of it. So what analysis will you carry out? This technique is a very very important technique when you're going to learn about environmental microbiology. um, do you have any idea what that technique is, what that analysis is? The study of whole microorganism in a given sample, to make it very simple. Again, this is also omics technique only. Okay, that's perfectly fine. No, I'm not asking about any single experiment. I'm just asking about a whole analysis that you're going to do, or the study, basically. Like epigenetics, we told a study of environmental effects on gene, something like that. All this can be a part PCR, 16S, yeah. But what the whole study is called. I again told you it's kind of omics technique only. No, we don't use generally it's not chromatography. Again, chromatography is a single experimental technique that's used. I'm asking about the whole study. Okay, those were great guesses. Let me tell you, it's called metagenomics. So, as you know, omics stands for the study, and you're going to study about the gene basically. Meta stands for all the organisms. So, when you're going to say metagenomics, it means that I will give you an environmental sample, and you're going to study all the microorganisms that are present. You know neither of it. I have not told you even a single clue about okay, this may be present or that may be present. You just don't know anything about the sample. You can do a metagenomic analysis and find out like basically what microbes are at least present in that. Metagenomics can be done to any sample. So probably I give you a 10 rupee note, and you can uh, perform a metagenomic analysis using that note to find out what are the microbes generally present in that one uh, rupee note. Okay. Uh, or a coin or any surface or a soil sample or a water sample, anything basically. So you are going to carry out few steps and find out based on the genes present in that, you're going to find out okay, these organisms may be present, and you get a whole cluster of organisms that were present in the sample. Okay. And final question related to this study is basically what does a methylome or a methylomics refers to? So now we know genome mix is a study of gene. Proteomics is going to be study of proteins in organisms. Transcriptomics is going to be study of the transcripts, the RNA transcripts. So basically we call RNA as transcripts. So transcriptomics is going to be the study of RNA. When it's going to be metagenomics or metaproteomics, metatranscriptomics, it's going to refer to the whole sample that you have, all the organisms that are present in the sample. Similarly, what does methylome or methylomics refers to? Right, perfectly right. So this is a kind of a confusing term because when I say proteomics, it's going to be study of all proteins, but methylomics is not study of mm, methyl groups that are present. It's the study of methylation. No, it's not study of the distribution of genome. It's a study of the complete set of DNA methylation modifications of a gene. So in epigenetics, we saw right. Uh, DNA methylation is one of the key factors in epigenetics, basically, because turning on a gene and a turning off of a gene are very very important in terms of what proteins they produce, and what kind of metabolism they're going to carry out based on the presence or absence of those proteins. Okay, so in that case, methylation is a very DNA methylation is a very important step, and the study of all this DNA methylation we collectively refer to as methylomics. Okay, so here let me explain what metagenomics is. So we have seen that metagenomics is very important and stuff. So for example, let's take this example. This is a very pictorial representation of a stomach. Okay, so you have multiple microorganisms here. What you do is you collect the sample, you extract the DNA. Again, what you're going to do is sequence it. Finally, you will get the metagenome. Okay. Similarly, you will have multiple samples, like you have that sample and there are multiple microbes, so you will get a very big data set here. What you do is you're going to process this data set to find the genes present in it. Individually, you will be getting different genes. Okay, so based on that, what you do is you can identify what microbial species will be present in that. So based on genes, you can also find what are the functions and pathways that are being that can probably be in that microorganism. Okay, so using this metadata, what you can do is you can get biological interpretation, you can do a biomarker discovery, you can also have a complete data set uh, that is perfectly analyzed and verified. So based on that, you can have microbiome based prediction tools. You can do a clustering analysis, you can do ecological modeling, phylogeny reconstruction, strain tracking, and epidemiology as well as population genomics based on this. So here in this case, since we have this human microbiome, you can do the epidemiological studies and population genomics also. So that is how important this metagenomics is. So basically what you do is you collect your sample, do the DNA extraction, do the sequencing, you get the metagenome data, and based on the genes present in that, you can find what proteins could be probably be expressed, or what micro our purpose, sole purpose as environmental engineers would be to know what species is present there. So this is what metagenomics is about. So this slide, I think you have seen in the lecture PPT. So this is very important to know what each means and to know what analysis you will require at what time. So if you need a DNA information, you go for genomics. If you need RNA information, you go for transcriptomics. Each have its own significance, because in the case of DNA, there is no guarantee. You find a gene, but there is no guarantee that it's going to be transcribed into RNA. Only when it is going to be transcribed into an RNA, it is going to be expressed as a protein, and here there is going to be the function of that protein in that particular microorganism that's going to confer some property to it, right? So genomics data is important in its own way. Transcriptomics data is also again important, and this proteomics, translatomics, and interactomics are very very important to know the diversity of proteins that are present in our microorganisms, which will confer its structure, and it will also confer function to the microorganism. So this is very important. Okay, and then you can also study about the metabolites. So that is called metabolome or metabolomics. In the case of, if you're going to study the entire microorganism that's present, it's obviously going to be called as microbiome, virome, or mycobiome, based on what microorganism. It's going to be virus, or it's going to be fungi, it's going to be algae, whatever it is. So based on that, your studies will depend. This is very important to fix your purpose. Fix your purpose and find the perfect tool, or the perfect analysis, or the experiments that you need to do that particular study. So this is the next question. So what is a technique that is used to generate alignments between a nucleotide or a protein, that is usually referred to as a query, and a nucleotide or a protein within a database? And the nucleotide or protein within a database is referred to as subject. You have two things. You have a query and you have a subject. To generate alignment between these two, what's very commonly frequently used? Okay, to make things a bit simpler, this question is based on a bioinformatic approach basically. So whatever we have discussed so far was some kind of a omics technique, PCR technique, molecular biology, everything was involved. This question is purely kind of a bioinformatic question or a computational technique basically. Okay, so any of you here have any idea about bioinformatic techniques, tools? Sorry, tools basically, not even techniques. If not, like I'll just uh, go through the questions that are related to it a bit faster. If you have no idea of it. Okay, have any of you heard about blast? So the answer for this question is blast. This was again discussed in one of the lecture videos extensively. Yeah, right. Cluster is also a bioinformatic tool basically is. Uh, do you have an idea what blast means? That's my next question. So blast stands for, we say we use blast, we perform blast for analyzing or doing a bioinformatic analysis. So what's blast? Okay, right. As you have mentioned, blast is a basic local alignment search tool. So what you are going to do is you're going to do perform alignment of the DNA sequence that you have with the already available sequence in the database. So based on that, you can find out for example, I have some kind of a nitrobacter with me, which was already studied very well. I have isolated it from some unknown sample, from a water sample or something, or soil sample. What I did was I did the sequencing and I found the sequence of the bacterium or the microorganism. I don't even know what that is. Okay, so I basically have the sequence of it with me. Now, in order, before going to any other analysis, I should first go to the database that is already available and check if that particular sequence, DNA sequence, is already recorded or established. So, if that is recorded, my work is easy. I will just know what organism is it. Right. So for that, what you do is you go to the NCBI blast tool. NCBI offers this blast tool. So you go to that and input your query sequence. It is going to perform the search with all the sequences that is present in that database and going to tell me, okay, whatever you have input is the gene or the genome of a nitrobacter. So what you have is a nitrobacter. it's confirmed. If it is not available, I am just going to report that this is not available, and I'll report it as a new organism. Similarly, when you have a part of a gene and you have you can just input that gene and know uh, what kind of a gene it is or for what it could potentially encode based on the available available database data. Okay, so these are the uses of blast. This is why we generally perform this blast. There are 5 different types of blast searches as we see here. So we know we have a query sequence that we are going to input, and we have the database subject. Okay, these both are going to be compared, and we are going to be given the output. Okay, so in blast n, what happens is I'm going to input a nucleotide sequence, for example, a DNA sequence basically. And the search is also going to be performed against nucleotide sequences only. Okay, so this is what is called as blast n. All these types are based on what sequence I'm going to input, and to what it is going to be compared in the database, to what sequence it is going to be compared in the database. I give a nucleotide sequence, it is being compared with other nucleotide sequences in the database, and the output is given to me. This happens in blast n. In case of blast x, what happens is I will be giving a nucleotide sequence. The tool itself is going to convert it into a protein sequence. So it's very simple, right? I have a DNA sequence, then you convert it to RNA sequence, and basically, and then you convert it to a protein sequence. It's based on the amino acid codes, the triplet codon, and the amino acid codes basically. Okay, so it's going to do the work, and it's going to compare my nucleotide sequence against a protein database that is present. So what it has done is it has converted it into a protein sequence, and going to compare it with other protein sequences, and it's going to give me the output. This BLASTX is generally used for identification of potential proteins encoded by query sequences, or detection of novel viruses. So these are the general purposes of it. Okay. Next is the blast p. Blast p is very simple. Like blast n and blast p are very simple. In blast p, I'm going to give a protein sequence. It is going to be compared or searched against other protein sequences that are present in the database, and this is used for sequence identification and similarity searches. T-BLAST-X is one thing where I give a nucleotide sequence. It is going to be translated into a protein. The database is also searched against a nucleotide only, which is again translated into a protein. So both my query and the database are nucleotide sequences which are translated to protein sequences. Okay, this kind of analysis is used for the identification of nucleotide sequences that are similar to the query based on their coding potential. I have a sequence. For example, we have this code degeneracy and other stuffs where two different codes, slightly different codes, can code for the same amino acids, because we have degeneracy in our codons, right? Only um, uh, two of the amino acids are very unique, that only one triplet codon is going to code for that amino acid. In other cases, more than one triplet codon can code for the same amino acid. So in that case, what happens is, though my nucleotide sequence has a minor modification in it, final output, the protein is not going to change much in the properties. So for this purpose, we use T-BLAST-X, and then we go for T-BLAST-N, where I input a protein sequence. The database is a nucleotide translated to protein. Okay, we have a nucleotide sequence. I have protein sequence at the end in my database. So, but that protein sequence was obtained from a nucleotide sequence. Okay, it is interpreted in the nucleotide sequence is interpreted into a protein sequence, and this input protein sequence is searched against that database, and finally my output is given. This is basically used for identification of database sequences encoding proteins to the query. Okay, so this is what the entire concept of BLAST is. BLAST is generally a search tool that's used for genomic identification or analysis, and these are different types of blast. And this is a very basic and a very useful tool in our microbiological analysis. Is that clear? Any doubts? Any doubts, or shall we proceed? You can give me a response or a thumbs up if we can proceed. Okay, okay, right. So this is the next question. So this question basically deals with the microarray technique. Okay, so we have seen about sequencing, we have seen about blast, we saw about PCR, all of it's done. So now it's about microarray. Okay, there is one response. I'll also wait for a couple of minutes more. Okay, we have two answers. Let's see which one is right. So this is the right order. We perform sample preparation, cDNA synthesis, hybridization, scanning, and analysis. So basically, let me tell you what a microarray means. I just have a slide on that. Uh, what you do is we have some cells. Uh, this this example is again for a eukaryotic system, not for microbiological system, but still, just to make the concept clear, we have different cell types. We culture them and we do the RNA isolation. So here, as mentioned, we have cDNA synthesis, right? What we do is we take the RNA. In case of RNA, what's going to happen is we have a DNA genome, but only those are that are going to get translated are going to get transcribed also. So at the end of transcription process, not the entire DNA is going to be converted into RNA. So we'll get only the genes that are going to be expressed. Okay, so that RNA is gotten, and what you do is you get the cDNA. I'm sorry, we get the cDNA. So cDNA synthesis is performed. cDNA is called as a complementary DNA synthesis. So you have a RNA strand, and when you are going to get to one DNA from it, it's going to be complementary to it, right? Obviously. So that is known as cDNA synthesis, and then you are going to hybridize it. So this is a microarray chip basically. So in this you have multiple wells. You are going to hybridize uh, that onto this microarray, and then uh, we are going to scan and analyze. So in this case, what happened is we have gotten different RNA isolates, reverse transcription. When you So generally for the cDNA synthesis, what you do is something called a reverse transcription. Okay, um, transcription, as we all know, it's RNA coming from a DNA fragment. So your template will be a DNA, and finally after the transcription what you get is a RNA. As a name suggests, reverse transcription is the reverse of that. You have a RNA fragment, so from that if you're going to get a DNA, this is known as reverse transcription process. And there is also something called a reverse transcription PCR that gives you multiple copies of this complementary DNA or the cDNA at the end of the PCR process. So in that PCR process, basically the reverse transcription is also going to take place, and the amplification is also going to take place. Okay, that's something uh, that was just another additional fact. So here what we do is we get the reverse transcription and we do the fluorescent tagging. The fluorescent tagging is done differently to uh, the gene, based on the genetic differences. Okay, now we are going to hybridize on a microarray and we are going to detect it. So we are going to see it with the detector. What happens is, in whichever there is no cell, or the no, where nothing is present, you get a gray signal. In the normally functioning cells, because the normally functioning transcripts, what happens is, this green color fluorescent tag is going to get bound, so you get this green signal. When there is going to be some modification in it, what happens is red color tag binds and you get a red signal. Whatever is present in both, you get in this yellow signal. Okay, because it's going to be present in both the cells. This example is generally based, this concept is generally explained based on a cancer cell. For example, you take a healthy cell, you take a cell that is has cancer in it, okay, and you do this RNA isolation. So in normal, compared to the normal cell, there will be some genetic difference in the cancer cell. Some other proteins will be expressed or something like that will happen, right? So based on that, you do the fluorescent tagging, and when you see here, you can see that this yellow color, this green color will be the normal cells, whereas the red colors will be the pathological or the cancerous cells. Okay, so this technique is generally used to study the differences. Based on this, you can take the gene out and do the genomic analysis, transcriptomic analysis. You can sequence it and you can do further analysis based on whatever your study purpose is. So this is the DNA microarray technique. Here the analysis is done once you are going to hybridize it to a microarray. Since you have multiple small compartments here, this technique is known as a microarray technique. Oh, okay, with that we go to the next concept, gene transfer concept. We have two types of gene transfer. So the gene transfer from one bacteria to another, that is not its offspring. We have bacteria A and bacteria B. There is a gene transfer from A to B, but the B bacteria is not the offspring of A bacteria. Then what is this phenomenon called? It's not conjugation basically. Conjugation is a type of it, but the phenomenon. I'm asking about the name of the gene transfer phenomenon. Conjugation is partly right, but it's not the exact name of the phenomenon here. Right. It is known as horizontal gene transfer. Ah, right, HGT. So now you can answer. Horizontal gene transfer can occur by Generally, we discuss about only three things, right? So I have given three blanks. Great. That's the right answer. It's conjugation, transformation, and transduction. So this is what horizontal gene transfer is basically. You have a donor cell and a recipient cell, and the gene transfer happens, and there are three methods of it. I think I have a slide at the last for it for transcription, transduction, and conjugation. We'll look into it. Now you can match the following. This is one question based on genetic mutation. You can just give me A1, B2, C3, something like that in the chat. Two responses that say A2, C3, and B1. That's perfectly right. It's A2, B1, and C3. Again, one more question related to mutation. You just have to again match the mutagen based on what it does. Okay, I think there are different answers. Okay, let's look at the answer for this. It's A2, B3, and C1. Okay, acridin is kind of an intercalating agent. So it intercalates between DNA, in between the DNA double helix. So in that way, it is going to cause the mutation. Whereas, as a name indicates, 5-bromouracil is a base analog. Base analog, as a name suggests, is something analogous to the given base, but it's not the original base, so there is going to be mutation happening there. So it obviously 5-bromouracil is going to be the base analog of uracil. Okay, you have a bromo group in it, and it's going to act as a base analog where it's not the actual nucleotide that has to be present at that point, but it's present and it causes a mutation there. Okay, and the third one is uv light. Uv light, what happens is when uv dna is exposed to uv light, thymine dimers are formed. For example, a thymine thymine bond is formed. This is called a thymine dimer, which generally doesn't occur in dna. Okay, so acridin is an intercalating agent. Another very famous intercalating agent is ethidium bromide, which we use in case of some gel running and all. So acridin, ethidium bromide are all intercalating agents. This 5-bromouracil or a 5-chlorouracil, they are all base analogs. UV light generally causes thymine dimer formation, which causes the mutation in the genome. Is that clear, or any doubts in it? Okay, so that's it. As we saw, 5-bromouracil, aminopurin, all these that have this name of this bases are going to be acting as base analogs. Okay, so this bromouracil is going to be incorporated like T, occasionally faulty pairing with G, and incorporated aminopurin is incorporated like A and faulty pairing with C. Chemicals that act, which I haven't mentioned in that question. So my question was based on base analogs, intercalating agents, and radiation. One more kind of mutagens are chemicals. So for example, nitrous acid, what happens is nitrous acid deaminates A and C. Okay, and hydroxylamine, it reacts with C. And we also have some alkylating agents, ethyl methane sulfonate, mitomycin. All these, what they do is, in the case of monofunctional thing, they have only one single function, that is they put methyl on the G, or they cause parity pairing with T. In case of bifunctional, they do two functions, that is cross linking of DNA strands, and the faulty region is, what happens is when this cross linking happens, this is excised or cut by the DNA enzyme, which results in some point mutations or deletions. In the case of intercalating agents, as I told, acridins and ethidium bromide can be used as the intercalating agents. So they are going to insert between the two base pairs. So you have a long DNA strand, you have a stacked kind of arrangement, right? So in between two base pairs up and down, this intercalating agent, as the name suggests, is going to bind and cause insertion or deletion there. Finally, coming to radiations, we have ionizing radiations as well as ultraviolet radiation. So this ultraviolet radiation, what happens is pyrimidine dimer or a thymine dimer formation. It may lead to some error or deletion. Similarly, ionizing radiations, what happens is they do a free radical attack or break the chain. This may also lead to mutation. To something that we saw previously, um, a mutation that causes a faulty protein is a missense mutation, or an incomplete protein is a nonsense mutation, and a normal protein, there is mutation in the gene, but still there is no difference in the amino acid that has been translated. So this is known as silent mutation. Okay, so the last question for today, as I told in horizontal gene transfer, I have this one more question related to that. In transduction, the genes are transferred from the donor to recipient by. It's a very simple question if you know the concept of it. by the end of this question, we will just discuss about what's conjugation, transformation, and transduction and close today. Okay, there is one answer. Okay, that's right. Phages, bacteriophages, phage virus, however you call them, they facilitate this transduction process. So let's basically see what's the difference between conjugation, transformation, and transduction. So in the case of conjugation, what happens is there is a donor cell and there is a recipient cell and there is a physical contact between these two cells. So the physical contact between these two cells happens by the means of pilus here. So this is again a protein or a tube which renders the connection between these two, and one of the DNA strands from this bacteria travels through the pilus to the recipient, and this gets this new plasmid for the genes that are present here. So this is basically the conjugation process. In transformation, there is no such attachment and all. It's just there is some dead bacteria. It releases its DNA fragments. The DNA fragments are as such present in the environment. There is some other microorganisms that is present in the same environment. What happens is this microbe uptakes the free DNA fragment that is present in the environment, and that fragment becomes a part of the genome of the recipient. So this phenomenon is known as transformation. In conjugation, we have a physical contact in the term in terms of pilus protein. In transformation, there is no physical contact. It's just the uptake of free DNA fragment from the environment. And finally, in the case of transduction, what happens is we have bacteriophages that infect bacteria. They release their DNA because, as we know, virus don't have their own machinery. They just use the machinery of the bacteria to make multiple copies of their genome. They can also make the bacteria produce the capsid, other proteins that are uh, required to assemble the whole package into a complete virus again. So after that, what happens is they do this transcription, translation, everything with this bacterial mechanism machinery, and then finally these proteins and the DNA are again packed into this phage structure. These phages are released. So when they are released, they can uptake the bacterial genome also with them. When they're going to infect another bacteria, this genome may be released and this can get integrated into the genome of another bacteria. So basically, phages facilitate this gene transfer from one bacteria to another, and this phenomenon is known as transduction. So this is basically three mechanisms in horizontal gene transfer. Is that clear? Okay. So, and with that, this live session for week 5 on the topic microbial genomics is done. I'll just wait for 2 more minutes. So, if you have any doubts, you can unmute and ask, or you can put in the chat box. Okay. So with that, I have one more question for you. It is like I have seen some new people this week whom haven't, I don't remember seeing in previous weeks. So are you people okay with this kind of interaction session? Because this was how all the previous sessions were also carried out. If you want, you can just visit YouTube and you can just find the recordings of this previous sessions. So is this fine, or any changes or anything required in the mode of delivery? We generally have some questions I wait for the answers and finally we discuss the concept of that question. So that's what I did this week also. Is that fine? Right. Okay, so thank you all so much for your time and for interacting very well in the session. Hoping to see you. Um, YouTube link, I think it's there in the announcement section of the NPTEL. You can just type applied environmental microbiology NPTEL in YouTube, and you will get the page on my name, Guru Priya. You can access through that. Week 5 environmental genomic PDF. You mean which PDF? The one PPT that I have now, or the lecture PPTs in the actual lecture videos? Shagufta Khan? Oh, this lecture 5, this PPT, right? Oh, this I'll put it in the drive after the session only. I'll upload it in the drive uh, once the session is over. Okay. Okay, with that, thank you all for joining in. Uh, it was really great interacting with you all. So if you have any doubts, you can ask or else you can leave the session.